Good morning, everybody, and good afternoon. I'm really excited about this two-day program that we've set up for this workshop on multimodal data integration as part of the Novo Nordisk Foundation Center. We're sorry we're a little bit late, a couple of minutes late. We're having some technical issues with some links, so I apologize for that, and hopefully that will all be resolved soon. But the first speaker is ready, and we're very fortunate to have Melina Claussnitzer, who's Associate Professor at Harvard Medical School and an Institute member at the Broad Institute, and also a pioneer for variant to function analysis, and heavily involved in the Novo Nordisk Foundation Center. And she's going to speak on scaling variant to function for metabolic disease workshop on multimodal data integration. Thank you very much. Melina, just take it away.
Awesome. Thank you so much, Bjarni, and also Alex for organizing this workshop. Very grateful for that. And thanks for the kind introduction. I was asked to kind of set the stage. So the presentation that I have here is not going to contain any science. For that one next time, this is kind of setting the stage in the context of variant to function, what the Novo Nordisk Foundation Center really is about, and how we are hoping to develop data sets that are suited well for multimodal data integration efforts across the center and for the global and international community. So let me just start out setting the stage real quick. I guess the majority, this is not news for the majority of those dialing in here. We know that, you know, the era of genetic discoveries over the last 15 years have been incredibly successful at identifying these hundreds of thousands of genetic association with diverse diseases and traits in the human genome. The Novo Nordisk Foundation Center currently is particularly focused on metabolic disease, context of type 2 diabetes, obesity related diseases and traits, where we are looking at multiple thousands of these genetic hits that we are tasked to biologically interpret. One important note, and I'm going to come back to that in a moment, is that the majority of these hits are located in the non-coding part of the genome, where the interpretation is substantially more challenging than if we were dealing with primarily coding variation. So this enormous success of genetic discoveries provides us with this wonderful opportunity that we have started sketching years ago as kind of the variant to function framework, where we are looking at these genetic maps, genetic variation in the human genome that connects with diseases and traits across the human population, providing us with a tremendous opportunity and a foundation of really starting to think about extracting information about and cataloging, mapping, causal targets and mechanisms of action, which once we have done that would be actionable in terms of therapeutic discoveries and preventive strategies. Now, all that sounds wonderful. The big challenge here, and I'm going to just quickly stage a few of these challenges over the next few slides, is to kind of fill this gap and learn what are the mechanisms that connect these, robust genetic association of genetic variation on the one side and medicine or disease and traits on the other. Now, my team has years ago in 2015 introduced the so-called variant to function framework, or V2F in short, where we are, we have designed this kind of principled approach, combining experimental and computational strategies to establish causal relationship between the causal genetic variants at a given genetic risk locus, learn how these causal variants regulate regulatory elements, which target genes and proteins they control, and in which cell types and cell states they act, and eventually which morphological and cellular functions are under the control of these genetic variants and their effector genes, which are relevant to disease pathophysiology. And all of that, the consideration of disease-relevant context cell states appears to be particularly relevant. Now, This variant to function framework, has been applied across the community quite successfully. And I think, this blueprint from going from these genetic variants to their target genes and cells and pathways in certain disease context has certainly shown to be generalizable. The big challenge that we have been facing over the last year is to think about strategies, how we can scale that effort. And the way we have been, visualizing this challenge over the last year is this kind of layered network, where you can imagine that we are dealing with thousands of genetic variants, mostly non-coding genetic variants in the human genome, each of which can regulate any of the hundreds of thousands of regulatory elements that have been mapped so far. Tens of thousands of genes and proteins act in any of the thousands of cell states that have been annotated so far and control any given cellular function and biological effect, which, just highlights this kind of combinatorial challenge that we are looking at, and really requires us to think strategically and design principles of how we can start tracing these non-coding genetic variant effects throughout these different layers in this network, and inform targets and mechanisms of action that robustly link these mostly non-coding genetic variants to their underlying disease function. We have by now, over 640 million single nucleotide and indel variants across the frequency spectrum in the human genome mapped. Of course, only a fraction of those is relevant to metabolic disease, but it kind of, adds to this enormous challenge, combinatorial challenge we are facing, in order to traverse from disease genetics to disease biology. Now, we do know, and that's kind of the good news, that we have an intuition around the principles and the rules of molecular and cellular programs in human cells, where, we are dealing with these generic models, knowing about hierarchical relationships in the cell between molecular layers and cellular layers. From the dogma of biology, really, we know that, information flows across these molecular and cellular networks, starting off from DNA to RNA to protein, of course, and then eventually converging or modulating some type of morphological and cellular phenotype as kind of natural integration points for effects from many, genetic effects in the cell. So, staging kind of this, we are looking at this tremendous success from the era of genetic discoveries, and we are tasked to move that to the area of mechanistic discoveries. I just want to spend a couple of minutes to highlight a few of the major challenges that really confound or has been very confounding in terms of traversing the path from disease genetics to disease biology. The first is that and I kind of touched upon this already. We do know that common diseases, including metabolic disease, are highly polygenic. We also know that, the majority of these diseases and traits involve thousands of independent non-coding causal variants that are inherited along with their variants and high linkage disequilibrium. So given, due to the haplotype structure of the human genome, a given lead SNP that is identified and connected with a certain disease or trait is not necessarily causal, because it is inherited with dozens of other variants in high linkage disequilibrium. And as I mentioned, the majority of these variants is located in the non-coding part of the genome, where it is substantially less straightforward when thinking about variant interpretation compared to coding variants. Now, a second confounder is the typical sparsity of data sets. I mentioned, we are working under this model of hierarchical relationships in the cell. There has been different efforts over the last years put on, the interpretation of these different layers in the information flow in the across the cell. But to just highlight what I mean with this, we have a really good way of by now at mapping chromatin accessibility and gene expression signatures, even at the single cell level, and in a pretty impressive scale. Less focus, so has been put on developing strategies that allow us to extract at the single cell level more downstream phenotypes, morphological and cellular effects that are relevant to disease progression. So this is certainly, in our view, quite important to think about. And my team has been particularly interested in developing image-based profiling technologies that are informative for these disease relevant processes. And then most importantly, very rarely is it the case that we have for given cellular models that are relevant for diseases and traits. We have a comprehensive data set. What do I mean with comprehensive data set? Ideally, we would like to develop a foundational data set for a given cellular model that really allows us to systematically integrate data in a principled approach. And such a principled combination of large data sets would involve the context of a lot of different disease relevant stimulatory conditions and compounds, since we do know that these non-coding genetic variants act in a highly context-specific manner. We need, thousands and tens of thousands of perturbation. And when I talk about perturbations here, I mean both natural genetic variation as well as engineered variation in these cellular models. And then I'm touching upon what I just mentioned before in terms of this, capturing the information flow in the cell. We need a diversity of different readouts that are poised to capture these stochastic events through single cell mapping of different molecular and cellular effects in the cell. Now, another component of really enabling this is to think about, which cellular model system are we actually dealing with? From our perspective in my lab, actually the first few years, we have primarily focused on thinking about how do we benchmark high fidelity model system for metabolic disease that are really where we can confidently or have evidence in hand through stratified RD score regression analysis, heritability partitioning, and so forth, that the cellular models we are working with are actually informative for the pathophysiology of disease. So this is certainly a challenge that needs quite some thoroughness to it. And this is, why we are here in this particular workshop, is to develop and think about, what are gaps in the community, what are opportunities, and how can big data generation efforts work very closely with the machine learning community to develop the next generation of models for predicting the effects of genetic variants in the context of this kind of variant to function framework, and really have ways to predicting the effects on regulatory elements, target genes, cellular functions, and so forth. Now, for the Novo Nordisk Foundation Center, we have, in a nutshell, designed, a data generation effort in close interaction with many across the center of, designing certain massive data generation efforts. In the context of, or applied on metabolically relevant cellular models, and the massive data generation efforts that we are kind of tackling these days are large stimulation and compound screens in these high fidelity cellular model system. Then natural genetic variation, where we are systematically probing in pooled screens how natural genetic variation impacts molecular and cellular functions, and then of course the pooled massive CRISPR perturbation screen to really be able to measure these dynamic cellular programs through single cell readouts and allow for these global predictions on this path of genetic variation effects down the line to disease pathophysiology. The different cellular models we are working with is, we have a tier 1 set of cellular models that we feel most confident about. Because we have put years and years of effort into them in order to benchmark them. And these are the subcutaneous and visceral adipocyte model systems, along with multiple other model systems that certain contributors and teams in the Novo Nordisk Foundation Center have been worked on so hard over the last years. The criteria we are applying is, I mentioned high fidelity, benchmarking high fidelity model systems. So we have, particularly cared about, are these cells able to recapitulate cell state transition, differentiation dynamics, response to stimulatory conditions that are relevant to metabolic disease. We also, for adipocytes and those who are familiar with my lab's research know that we have developed this population scale cellular biobank of adipose-derived mesenchymal stem cells derived from subcutaneous and visceral adipose tissue in order to be able to study natural genetic variation in a highly cell context dependent manner in these primary cells derived from hundreds and hundreds of different donors. We also have benchmarked the system to be amenable to high-throughput CRISPR perturbation screens, and of course we have robust protocols available. Now, what are we doing with this cellular system? We are applying, as I mentioned before, these kind of 3 different massive data generation efforts or perturbations to them. The one is large stimulation and compound screens. The second is these natural genetic variation screens, where we are pooling primary adipose-derived mesenchymal stem cells from dozens of individuals into the dish, and then use sequencing, 10x multiome as readout, and image-based profiling as readout, and complement that with CRISPR perturbation screens, where we are using mostly the DCUS9 CRAB repressor system to systematically perturb not only the predicted effector target genes, but also the associated GWAS-associated regions to really establish causal connections here. And then, as mentioned, use these different readouts that are complementary in nature, where we are hoping that they're complementary enough, but also comprehensive enough to capture a large amount of information in the cell through 10x multiome and image-based profiling, where we are having an eye on protein level profiles as well. And as a result, we are dealing with these thousands and tens of thousands of different features per phenotypic modality and per individual perturbation that we are hoping to inform disease biology in this context. Now, we are particularly aware that we have to map how cells behave in the context of cell activation and differentiation, in the context of response to a diversity of different stimulatory conditions. I had mentioned this before. It is highly relevant in which cell, in which cell state a given cell is when we want to read out how natural genetic variation impacts that, particularly in the context of metabolic disease, where hormonal signaling and nutrient activation and so forth is absolutely critical. Now, I have to close soon. I just want to leave you with 2 slides. The one is kind of a snapshot of where we are now from the Novo Nordisk Foundation Center in terms of the adipocyte screens that we have put into place. This is a super exciting moment for us, because we have, spent years now to develop these pooled screens that, systematically interrogate how natural genetic variation acts on different molecular and cellular phenotypes, but also be able to match this with large stimulation and compound screens, as well as these CRISPR perturbation screens in the same model systems to develop this comprehensive foundational data set. We are right now at the very moment of, I would say, an inflection point where the systems are in place, and we are starting to really, what you're looking at is the data set from 2 weeks ago, a matrix of in total 1,600 stimulation and differentiation state and 150 natural genetic variation or donor context, and in total 36 million cells that are sequenced or imaged through image-based profiling and 10x multiome. And that will scale prospectively quite heavily because the systems are in place now. Now, I will close with just giving, this is a very full slide. I apologize for it right away. I just wanted to highlight, and we can, Alex and Bjarni, if we want, we could come back to this slide at any given moment later throughout the workshop, which is kind of summarizing, the different streams and thought processes that we have to go through and really tackle as a community you know, first line processing of the data, developing and plugging in already established data standards of certainly folks that are speaking as part of the workshop here have been have pioneered a lot of these standards and data integration methods. We are, of course, very interested in integrating these data with the GWAS data and running, you know, of course, SNP enrichments. Then the integration across these modalities, right, where we are now not only focusing on gene regulation, but also folding in more downstream phenotypic events. We have also, you know, started thinking about, okay, what about data integration where we derive data points from different technologies, for example, 10x multiome and share-seq, which is, you know, not trivial and quite challenging. And then really think about, are the data that we are generating currently sufficient to fuel these statistical and machine learning models, or do we have some kind of gap? Do we need to think about adding in more modalities into these efforts? Now, a few challenges, just real quick. Of course, the question is, you know, in terms of developing the next generation of models and looking at this kind of multimodal data set for harmonized data set for individual cell types relevant to disease. What are the right models to go after? How do we enable model interpretation, since we are really interested primarily in the interpretation of biological effects in the context of metabolic disease? You know, how do we develop and how do we think about the fact that the prediction accuracy of current models often is, you know, tailored towards accuracy high for molecular features, but how does that translate to actual individual single variant prediction? You know, and there's a few other challenges that we probably go through in the discussions along these talks. Of course, the context matters. Then in these models, how do we account for the fact that the distance to transcriptional start sites is often a confounder? Yeah, and I want to, you know, just thank the Novo Nordisk Foundation Center, the Novo Nordisk Foundation itself, of course, but also the center, everybody who has chimed in here in developing these, you know, framework and thinking about the strategies that we have at hand right now. And I'm super excited about this 2-day workshop and start discussing challenges and opportunities. The majority of the speakers will focus on variant interpretation in the context of gene regulation, but also Caroline, who will start now, and I think Alex Fabiani will introduce Caroline. But, you know, I just want to mention that this kind of connection of morphology and regular gene regulation is particularly intriguing here as well, where Caroline has focused a lot of energy on over the last years. So shout out and thank you again. And I think I hand over to, I don't know who.
I can take the baton from you, Melina. Thank
Awesome.
you so much. Can everyone hear me okay? We didn't do a full AV check. Yeah. Thanks, Melina, for that wonderful introduction. It really sets the stage for the kinds of things we want to be thinking about. And I'm very excited for this whole workshop.